All right. Any questions about Wednesday's material? Nobody? Okay, great then. In the chat box, go ahead and tell me where the energy, electrons, and carbon come from for a photolithoautotroph. I'm going to need 3 answers. Where's the energy come from? Where's the electrons? Where's the carbon? John, yes. Sunlight, inorganic, and CO2. All right, so let's get back to our presentation and take a look at some of these things. Share this. All right, so here again is your cheat sheet on that. Some interesting stuff comes up. Notice the 3 question marks there for photo-organo-autotroph. So something that uses sunlight for energy, uses organic sources for electrons, and uses CO2 for their carbon. I don't know and cannot find, and Wikipedia doesn't have, and the textbook doesn't have, any examples of this in nature. You think of why. I mean, maybe they're out there. Wikipedia didn't have photolithoheterotroph either, but Great Salt Lake is filled with those things, halobacterium. So maybe there is something, but I can't find it anywhere. What's weird about photo-organo-autotroph, and why isn't it present?
Isn't it that part that says inorganic?
No, it's an organo.
Oh.
Yeah, this one here. So something that uses sunlight and an organic source for electrons and CO2 for carbon.
Is it because that when they have sunlight, they still need sunlight for their energy, but they also need organic stuff, so they need to, is it they need to eat food for their oxidizing or for the electron thing, or is that the energy source?
Yeah, I mean, they do have to get it from an organic source somewhere. I mean, but there are weird bacteria out there we'll see that, you know, can live in nail polish remover, acetone.
So wait, when I read that, I kind of think of the Venus flytrap. Is that maybe along the lines?
Oh, I see where you're going. No, those are actually a photolithoauto. They're not getting their carbon from the insect. They're getting nitrogen from it. They're still getting their carbon from CO2. Yeah.
Oh, okay.
Yeah.
So it's probably due to the process of like photosynthesis. Like I mean, there's a certain way that it works, and they get their carbon from the air, and then it's just kind of, I don't know.
You're real close. Yeah. So it is something about photosynthesis. It's like, here's a plant, photolithoauto. Okay. So they're using sunlight. Where does plant get its electrons?
Isn't it from the water?
Yeah, it's from splitting water. So solar energy breaks up H2O. And so oxygen's a byproduct of this, but then we also get that free electrons from that reaction. And so photosynthesis is really set up to have sunlight split water. And so that's litho. You don't have to get your carbon from the air like a plant. We've got weird bacteria. Actually, they're archaea. They're just called halobacterium. Really perverse name for these because they're archaea. But we've got halobacterium in the Great Salt Lake, all that pink stuff in the north half. They do photosynthesis, they split water, but they actually are getting their carbon from other sources. They're eating their carbon from organic stuff. So it's really weird, but it probably has something to do with no current organisms, at least that I know of or that anyone I've talked to knows of, have this process. Maybe it's just really thermodynamically inefficient. Okay. But for all the rest of these, we do have examples. Let's take a look at our tree of life, and we're going to zoom in on cyanobacteria. Okay, right there. This is one of those major groups of bacteria. Here's a zoomed in view of that branch. This is a tree of cyanobacteria with showing a bunch of different species or genus complexes that we've got here. So they're broken down into these different groups, single-celled, or these like ones that kind of organize together, multicellular, or have these heterocysts, like actual cellular differentiation. Yep, there are multicellular bacteria, including, we're going to zoom in on this guy, nostoc. So this is nostoc. It's a multicellular bacteria, makes really big structures. You can see it. It's a photolithoautotroph. So H2O is the reducing agent. That's where the electrons come from. Electrons are reducing. What is the product of photosynthesis for this organism?
What was your question?
What's the product of photosynthesis for this organism?
Oxygen and yeah,
Oxygen.
It's oxygen
Yeah.
and glucose.
Yeah. The splitting up H2O for electrons means that O is around. So we've got oxygen Roman free. Okay. So that's kind of the waste product of photosynthesis. So this is a weird bacterium. How are labs working this semester? You guys going into class for labs, or
No, they're
Our
online.
lab's online.
they're online? Have you seen Nostoc yet? I know it's one
Yeah,
that you
it's
do
a
look at.
cyanobacteria.
Yeah, okay.
We learned about that last lab.
Gotcha. So you've seen this before, and you know that it's actually genuine multicellular organism with cellular differentiation. We've got like these little heterocysts that can fix nitrogen. Okay, so you've seen this. On a side note, how is online lab going?
My
Is it
last
working
term, I had an online lab, and it was kind of funky. This term's is a lot better. It almost
Okay.
feels like just another biology class. It's just another lecture, but it's presented in a different way.
All right.
So they stack on top of each other almost.
All right. Well, I hope it works out. That can be tricky. I've never taken an online lab before. All right, so that's Nostoc. And let's just zoom over, get to meet another one here. Proteobacteria. This is a giant group of bacteria. Here's a tree of the different proteobacteria groups. Alpha, gamma, and delta are the big ones. We're going to look at gamma proteobacteria. So if I zoom in on that monophyletic group, it expands, and we got all these different named and described species in here. Some of these you've heard of, Pseudomonas, right? But let's go over here into the basal stuff and just expand on this little group, because we got some weirdos hanging out. Like this one, the purple sulfur bacteria. All right, this one is not oxygen tolerant whatsoever. Can't be around it. It gets its energy from light, so it's a phototroph. Its reducing agent is dihydrogen sulfide. Okay, so it's a lithotroph that doesn't have carbon in it. And it gets its carbon source from CO2, so it's an autotroph. What's the product of photosynthesis for this one?
Shouldn't it be sulfur dioxide
It's sulfur.
or
Yep,
just sulfur?
yep, S. And S2 is what it combines to form. So yeah, sulfur is, I mean, obviously purple sulfur bacteria. So these are stinky. All right, where would you look for these in nature?
Near hot springs and I imagine volcanoes. So are they an extremophile?
They are an extremophile. So, I mean, yeah, dihydrogen sulfide, anyone know? Yeah, that's volcanic largely in origin. Okay, so we've got like thermal vents, that kind of thing, but they're not oxygen tolerant. And so it's got to be an anoxic environment that also has CO2 and sunlight. So that's going to be limited to a few weird places on Earth. Normally, if you've got CO2 and sunlight, oxygen's probably around also, right? Because you're on the surface of the Earth. So these things live in like thermal vents, Yellowstone National Park, that kind of thing. Then we've got the purple non-sulfur bacteria, close relative, usually not oxygen tolerant. They're phototrophs. They are organotrophs and autotrophs. Okay, so these are the ones I hinted at earlier. They need an organic source of electrons. They need light and they need CO2. So a bit different from plants that want water for their electron source. If you want to grow these in a lab, you actually can. You can go to some soil. Usually you can find them kind of buried in there somewhere.
Would these be like yeast?
Like what?
Yeast?
Kind of like yeast. I mean, but yeast wants to eat your bread and your sugar. Can you hear my dog snoring? He's getting real loud, sorry. These want to eat things like nail polish remover. So acetone is something they really enjoy as an electron source. Okay, so you can fill up a jar with like boiled water, so there's oxygen's all gone from it, and seal it off real well with some CO2 in there and throw in some nail polish remover and they'll start turning purple. These purple non-sulfur bacteria start growing. So there's just weird organisms out there. The biochemistry, like the diversity is insane.
Why is it called non-sulfur?
Oh, because they don't use dihydrogen sulfide for their electron source. They use organic stuff like acetone.
So why is it called non-sulfur instead of like something else that?
Oh, like
Is
why
it?
is it not called purple acetone bacteria?
Yeah.
Okay. I gotta apologize on a whole for biology, just naming things kind of poorly sometimes. These are named purple non-sulfur bacteria because in the tree, they're right next to the purple sulfur bacteria that were named first. And everyone thought these were the same stuff, and so now we just call them non-sulfur because we know they're different.
Okay, thanks.
Yeah, it's pretty dumb. Sorry. And then one more weird bacterium, Aquifex. What can you tell me about this bacterial group based on this tree?
They're the closest, like they're the ones that were the first to evolve.
They're very old.
Technically, let's try to use proper terms here because not first to evolve by any stretch, but earliest diverging from the rest of the group. it may seem like a semantic difference, but I want to be real clear. So yeah, they are the earliest diverging. It's not that they're more closely related to archaea and eukaryotes, because they're not. They're sister to all these other groups. Okay. But they are kind of the weird, real early diverging group from bacteria.
So would they be kind of like considered an outgroup, or would it be
If you
different?
were, well, they technically are still bacteria, so you'd be careful about calling them an outgroup to bacteria. You could call them an outgroup to all the rest of these.
Okay.
The definition of an outgroup can change. It really is just the thing that you're not talking about that's closely related.
Okay.
So if we were drawing a circle around all these other bacteria and calling them some name, like the non-weirdo bacteria, this would be the outgroup.
Okay, that makes sense. Thanks.
Yeah. So we can get in there, take a look at Aquifex. It's one of these early diverging groups. And here's where it grows. It's very hyperthermic. It needs really, really hot temperatures. It loves oxygen. It hates dihydrogen sulfide. That'll kill it. It's a chemolitho heterotroph. All right, so knowing what you know about its growth conditions and what we talked about with our purple sulfur bacteria, this one's pretty weird. Pictured on here on the right, you can kind of see like these little gross snot trails hanging on the rocks in the water. That is Aquifex. Okay. Look at its energy formula here. It takes pure hydrogen and oxygen, and water is the waste product. Where does it grow? Not here in the left side. Why not?
Because it doesn't like the hydrogen sulfide.
Yeah, it doesn't like the whole hydrogen sulfide. It can't handle the hydrogen sulfide that's coming out here. But it needs to be really hot to grow. So this Aquifex organism is literally limited to like this stretch of 50 meters downstream from these like hydrothermal vents. That's the only place on Earth that it can grow naturally, where it's hot enough, it's got that hydrogen, right? And it doesn't have the H2S, and it's out in the sunlight. These are weird, right? Very limited distribution. Really a fun study for bacterial evolutionary biologists, because you've got like these populations that really have not been anywhere for millions of years, anywhere else. They're stuck right there. Let's move on over and take a look at an archaea. Please note on here that archaea is sister to what?
Eukarya.
Eukarya. Yeah. So an archaea is more closely related to you than a bacteria is. And let's take a look at this one. Methanococcus geneshii. Okay. It can't handle any oxygen. A single molecule can basically kill it. It takes hydrogen, CO2, and makes methane. This is a methanogen. It generates methane. And it doesn't get its energy from light. It gets it from chemical sources. So somebody classify its metabolism for me in terms of the major metabolic group names.
Chemolithoautotroph?
Chemolithoautotroph. Yep, absolutely. Where would you find this thing?
Probably in a mine,
Okay,
like
there's
near
going to be
petroleum.
oxygen there.
Yeah,
Underwater near vents or something?
It's got to be pretty deep because the ocean has plenty of oxygen in it.
I would think it would be in a volcano. Like, pretty. I don't know if it could survive possibly actually in the lava itself, but I know those produce a lot of hydrogen. I don't know if they produce a lot of carbon or CO2.
Yeah,
But-
they do.
Okay.
Lots of CO2, lots of sulfur, lots of hydrogen, but I'm gonna burst your bubble here. Nothing can survive lava.
Dang it.
If you
I'm
drop
guess-
something in lava, man, just forget it. It's gone. It's gone forever.
My guess is still like in oil wells and near that stuff.
Oil well type stuff. Yeah, these are commercially really important because methane is natural gas. That's a big commodity. I turn on my oven. That's natural gas right there, methane. Um, the bulk of our natural gas comes from like offshore drilling in the continental beds. So it's like buried underneath, meters and meters and meters of thick mud underwater. There's no light getting to it. It doesn't need that, but it's getting that CO2 and hydrogen and stuff kind of bubbling up. And so this is commercially important, and that's where we get a large portion of our natural gas. It's just kind of tapping these underground, underwater, anoxic environments.
So
We
why
don't
can't-
know of, one second, we don't know of any organism that can make methane in the presence of oxygen. As far as we know, it has to be anoxic. Yeah, what was your question?
So why can't they take these archaea and reproduce them in a lab to make major sources of methane?
Oh, you could, for sure. But, I mean, I think it's probably a cost effectiveness. Is it economical? It's obviously going to be cheaper just to go out there and tap what's already growing. I mean, we're talking like continental scale, right? Quadrillions of these things. You'd need a really huge building constantly going, keeping the conditions right, making sure no oxygen gets in. I think it'd be pretty expensive.
If they're tapping it though, aren't they exposing these archaea to oxygen?
Yeah, at
So-
first for a little bit, as it goes down, they establish everything, but then they're just trapping it essentially. That
Oh.
methane is just kind of like bubbling up slowly, and so they're digging down in there and building like a little hood trap to catch the methane.
That makes sense.
So maybe this is a little bit stupid, but I'm not sure exactly what to write down or what to pay attention to in the slides. Do we write down the name, or I don't know?
This is practice for you getting to know some of these groups. We're going to spend time throughout the semester, focused on, we're talking about bacteria now we're talking about plants, that kind of stuff. It's organismal biology. Right now is just an overview of the tree of life, getting to know a few of these and seeing if you can identify the metabolic group based on what I tell you about it.
Okay, so basically like the chemolitho-autotroph sort of.
Chemolitho-autotroph, and
Yeah, that
can
sort of
think critically about where this thing could or could not live, that kind of stuff, right? What, where would you not find this thing based on its metabolism? Because when we start looking at like biofilms and stuff, when you talk about bacteria, we'll see that some bacteria can't form biofilms together because their metabolic waste product is poisonous to the other one. So just to get you start thinking about it.
Okay, thank you.
Are
Not
there
to
any?
have- Memorize the names here.
Are there any
What?
organisms that can alter the metabolic group if they need to?
Yeah, those really bug me. They're super annoying. Very cool. Not easy to fit into categories. We may see some of those. The biggest one that we'll take a look at is plants, where sometimes they're a photolitho-autotroph and sometimes they're a chemo-organoheterotroph. Every plant out there does that. And now eukarya, everyone's favorite. Okay. It's a relatively small group compared to the others, and biochemically it's not super diverse. But let's go ahead and zoom in, take a look at eukaryotes. So we're going to learn about these in like 5 weeks or so. We'll spend a lot of time looking at like the protist groups, okay, amoebozoans and archaeplastidos and all that. Way over here, that's our animals, metazoa, all right? But let's take a look at Archaeplastida. This is our green plant and algae group. So we can zoom in on that and just take a look at land plants and zoom in even more and mosses. Okay. So we've talked about this one already. Photolithoautotroph. Energy, electron, and carbon. Where is it coming from? Energy is from the sun.
Sun.
Yep. Electrons are from
Organic compounds.
no
Inorganic.
inorganic, specifically water. And the carbon source is
Autotroph.
CO2.
Yeah, carbon
CO2.
dioxide.
Yeah, from the air. Perfect.
I forgot what classifies a material as organic or inorganic when determining between organotrophs and lithotrophs.
Carbon.
So if the reducing compound has carbon in it,
Yeah.
what's an example of that?
Acetone, ethanol,
Okay, awesome.
all the lipids. Yeah, all the so I'm sure probably most of you haven't had OChem yet, but if it's got carbon in the molecule, it's typically classified as organic, except for CO2. Okay. So CO2 is the like kind of default inert mineralized form of carbon, and it's technically, for biologists, considered inorganic. And so that's why autotrophs use CO2.
Sweet, thank you.
Now, two questions for you. Can plants undergo aerobic respiration, and can they be chemoheterotrophic? Well, I answered the second one for you. Said yes. What's going on with this? If they're photolithoautotrophs, what is the point of being a chemoheterotroph?
Diversity.
All plants do this.
I imagine it's kind of how they respirate. Like they take the chemicals that they've made and then they break them down, sort of. That's my
Yeah.
guess.
That's right. So, I mean, photosynthesis doesn't give you biomass. All it is taking air and turning it into sugar. You still have to eat that sugar. You still have to burn it and break it down and all that kind of stuff. So an animal comes along and it just takes the sugar. It takes these organic compounds and breaks them down. Plants are doing the same thing, but they make their own first. They take sunlight, they steal some solar energy and some water and some air, and they make sugar out of it, and then they eat the sugar. And so, yeah, when the light is shining, they're photosynthesizing and they're using solar energy to make sugar. At night, they're respiring. They're actually breaking down that sugar through cellular respiration. Okay. So plants are switching between these two metabolic groups. They do both.
So does that mean, that we're always told that we get our main source of oxygen from the plants, but they're also using the oxygen?
Oh yeah, they're definitely using the oxygen. Yep.
I've been lied to.
No, our main source of oxygen still is plants, but they're turning it over also. Yeah.
All right.
Plants got to eat too. All right. Now let's take a look at another weird eukaryote. This one breaks a few rules of our eukaryote preconceptions, probably. everybody coming into this class, for the most part, it's kind of a generalization, but I say eukaryote, and everyone's like, animals. I'm like, yeah, no. Right? Animals are a tiny part here. Let's take a look at this little group called Dictyosteliae. These are slime molds. Okay, pictured here. Spores fly off, and out of the spore hatches a little amoeba. Crazy.
Is it still just one big cell while they're doing that as well?
Right until the point where they make the little balls on a stick.
Wow.
Yeah, and so it can be a big, like basketball-sized cell, kind of acting as a multicellular organism, crawling around, responding to stimuli, all that kind of stuff. And then when it's like out of food or the weather changes, it's like, okay, things are kind of going to crap. I'm going to reproduce and go dormant. Then it breaks up into a bunch of single cells again and turns into spores. So
Is that one of the biggest cells we know of?
I think ostrich egg takes the cake. Yeah. But yeah, it's one of the larger ones for sure.
Would anything happen if you got that like on your skin or something?
No, you can even eat them. They fry up like scrambled eggs and they taste like nothing. I got my PhD in slime mold ecology, so I was fascinated by these things as an undergrad. And I was like, that's it. I'm going to spend the next 5 years of my life studying slime molds, and so I did, which means I also got to eat them. And then let's take a look at fungi. Another group here. Okay, these are pretty famous, right? They do a lot of biochemical hard lifting for us humans. Beer and bread are both from yeast. They are what we use to make ethanol for our gasoline out of corn stalks and switchgrass. They're the ones actually digesting grass in a cow's rumen so that a cow can actually break down cellulose. Cow can't do it, the fungus has to. Okay, so agriculturally, ultra important. We got zombie fungi that take over the brains of ants and make them kill themselves. Really cool, but we will spend more time in this class on plants, animals, and fungi than any of these other groups. Right? We'll talk about bacteria. We'll talk about all the protists together, so stromatophiles and slime molds and all that. We'll spend time on them, but I wanted you to kind of get to know a few of the groups that we're going to be seeing, talking about, and to keep in mind their metabolic diversity and their biochemical diversity, because it really shapes what they can do. Okay. If you are interested in metabolic pathways and who can do what, I highly recommend checking out the KEGG. You can just Google KEGG and like genome, and it'll bring up this KEGG pathway. It's a clickable website, so you can go through and say, okay, here's each one of these little dots is a known enzyme, a little protein that contributes in some way to transforming one molecule to another, right? So how is it that a methanogen takes some hydrogen and some CO2 and makes methane? Well, we actually know all of those genes, and we know in what order things are transformed. And so you can click on these and kind of follow it around. You can highlight pathways, and it'll show you what organisms we know have that pathway present in their genome, that kind of stuff. It can be pretty cool. Okay. I'll stop it there and switch to the final slide, which is, are viruses alive? Do they do any of this metabolic stuff that we've been talking about? Going back to what is life? Where do they fit in this whole tree of life thing? There's a question real fast while you're thinking about that. Can slime mold sense where bacteria are? Chemically, yeah. If the bacteria have chemicals or digestive enzymes they're putting out into the environment, the slime mold can chase those down along the chemical gradient. So what about COVID-19, SARS-CoV-2? What metabolic pathway does it have?
I mean, I don't know that it has any. It kind of just feeds off of the host.
I know
I
what
mean-
you're saying. I'm going to take
I
slight
don't know
issue.
enough about viruses to
Okay.
say.
Would it be
One issue I have is that you said it feeds off of the host, and
Oh,
technically
yeah,
it
yeah.
doesn't. It doesn't feed ever.
It reproduces through the host. That's
Yeah.
what I meant to say, sort of.
Yeah. And what about transposons? You know, the majority of your genome is virus DNA. It's true. You're more virus than human because of transposons. These are transposable elements in your genome. It's a section of your genome here, and all it does is code for an enzyme to copy itself and insert itself somewhere else in your genome. So when you transcribe that protein, all that protein does is copy that little section of DNA somewhere else into your chromosome. Is that alive? It's reproducing. It's growing. It can evolve. What's the other stuff about life? Stuff we've just been talking about, this metabolic notion. Okay. All the examples we saw, all the weird chemical metabolic reactions going on, they're there fending off chaos, entropy, right? Disorder. They're there kind of making sense out of disorder, ordering things for a little while with the input of solar or chemical energy, okay, whether it's geothermal or solar energy. We've got to keep feeding energy into a system to have it not go to crap, basically, to become disordered. So we had a big list of things we talked about with our what went into life. And I think we kind of agreed, I think most of us at least, agreed on metabolism, homeostasis, and cells. If anyone has an issue with that, let me know.
Didn't we later add on from what is life book said, where life is anything that organizes or has negative entropy?
Not anything that has negative entropy. Negative entropy is always going to be temporary because you got to feed energy into it. And once you turn off the energy flow, entropy wins. That's thermodynamics. That's how physics works.
So isn't it like whatever works against entropy, like actively sort of thing?
Yeah, it's actively working against entropy. I mean, but now this is going to be weird, but if you freeze water, it's actually more ordered than when it's liquid. So that's against entropy. So it's temporary, but I wouldn't call it alive.
Is that also to say that if you are, and then sorry, my connection's probably terrible, but if you're
okay.
heating up water, since you're adding energy to the system, would you, in that case, be in some minute way be adding to the mass of the system? More along the lines of thermodynamics. If it's pushing it back against entropy, would more energy be also more? Anyways,
maybe I got a bit confused.
I think I started it thinking it might have been more related than it was.
Okay.
Sorry about that.
So what are the arguments against virus being alive? They can't replicate and survive by themselves, and they need a host to replicate and survive. True. But so does a tapeworm.
Yeah, because isn't that the same argument? Like a lot of parasites, that's the same thing, and they're considered alive too. So
yeah, for sure.
Because like I've heard both ways about virus being alive or not, but I don't remember all the arguments against it being alive. Because we just talked about like a lot of the reasons why we could consider it technically alive.
I feel like the biggest argument against it being alive is this issue of metabolism. Right? Viruses don't have ribosomes. They can't make their own enzymes. Therefore, they can't catalyze their own biochemical reactions to stave off entropy. Okay. So for me, that's the biggest, or probably the best argument against viruses being alive. However, we have found more recently that viruses have things called ribozymes, which is RNA that's folded up into a funny shape that can work exactly like a ribosome and can catalyze reactions. It can be its own enzyme, which makes sense because we know that RNA can actually self-replicate without any other stuff going on. So I don't know. I don't think there's a right or wrong answer on this, but it's worth thinking about.
I've got a quick question. Do viruses have a mitochondria as well, or no?
No. They do not.
Do viruses grow from when they're young to when they're old, or do they stay the same the whole time?
Same size.
So would most biologists argue that viruses are not alive, or where do most biologists, or where do you sit on that?
I feel like they qualify as living. It's a self-organizing collection of proteins and organic compounds that has found a way to replicate itself using the environment that it's facing. To a virus, the bacterial cell or the human cell that it encounters is nothing more than another inert part of the environment that it can utilize. It gets even weirder when you start talking about things like transposons. These don't even have like a protein coat, right? It's just a piece of DNA. But what it codes for is an enzyme to make copies of itself. So if you're an enzyme, if you're a gene, and the code for you isn't to turn your eyes blue, but the thing that you make is to make more copies of yourself, you're going to be very successful. You're not worried about what color the organism's eyes are, whether they've got a unibrow or not, like some other genes, right? All you do is copy yourself. And so that's why the bulk of your genome is made up of this stuff. It's because it's really good at making copies of itself. So it's maybe one of the most successful forms of life out there. I don't know where I kind of fall on that. We do consider it like a virus, though, because it can't reproduce itself. It needs to harness our DNA polymerase to read itself, and it needs to harness our ribosomes to make the protein to get itself copied. Yeah. If you download this presentation, you'll find a video here on transposons if you're interested, or you can just Google them on YouTube to find them. But this concludes our introduction to the course. These first 2 weeks is kind of like, let's get to know what we're about to learn. Okay. Next week, we do have class on Monday, so quiz will be due before class Monday. And as usual, over the reading, and we're going to get into mechanisms of evolution and then just go through our groups. We're going to meet the animals, meet the plants, meet the fungi, and then see how they interact with the environment. All right, we're out of time. We've got our final candidate for bioinformatics professor today. So if you go to our general channel. You should find the link to that. It's at 1 o'clock if you're interested and can attend. If you want those bonus points, I need it written up by the end of the weekend, because we're going to meet and talk about these candidates as faculty. And I want your feedback before we discuss it. Any last questions?
I have a question.
Okay.
So on your website, it says the quiz is on Wednesday. So it's not on Wednesday, it's on Monday.
Wasn't that this week? Or
I
next
thought
week?
that was.
Do we not have?
We didn't have a quiz this week, I don't think. As far as I'm concerned.
Yeah, so quiz chapter 2 is due Wednesday before 11.
Mine is showing that it's due on the 25th.
If you look on Canvas, it says Monday, but if you look on the website, it says Wednesday.
Oh dear, that is a typo on the website. Let me share my screen so we can all see that. Yeah, because we do have class on Monday. And it is set to be due on Monday in Canvas. So I'll just get this changed. Thank you for catching that. Okay, so that'll be Monday, not Wednesday.
So is the quiz 2 topic, is that based off of the week 2 stuff, or is that off chapter?
No, that's chapters here. There's
Okay.
Yeah, so it'll go with that. So quiz 2 will be over this reading.
Okay, thank you.
Yeah, and sorry about the typo. That is Monday before 11. Go with what Canvas is telling you on that in that case. All right. Good chat here. Okay. I'll see you on Monday. Have a good weekend.
Thank you.
Yep.
Thank you.
Thank you.